So this development of placenta can be divided based on the days after fertilization. So it is divided into 4 phases. So what are these 4 phases? It is the pre-lacunar phase. Next is the lacunar phase. Next is the early villus phase. And the last is the villus phase. So, firstly, we have to understand that during fertilization, what happens? The egg and the sperm fertilize and it forms the zygote. So, once the zygote is formed, it goes on dividing. So, once it divides, it forms something called as a morula, that is a day 3 embryo. And once there is formation of the morula, there is something called as a blastocele formation, and then it gives rise to something called as a blastocyst, which is the day 5 embryo. So what is the blastocyst? It is again divided into the outer layer, which is something called as the trophoblast, and the inner layer, which is called as the inner cell mass, which is also called as embryoblast. So the trophoblast is also called as trophectoderm or the trophoblast, and the inner cell mass is also called as the embryoblast. As the name suggests, the embryoblast will give rise to the future embryo, and the trophoblast exclusively forms something called as the placenta. And embryoblast also gives some of the contribution, that some of the villus stroma, chorion, amnion, umbilical cord, and all arise from the embryo. But while the trophoblast exclusively leads to the development of the placenta. So once the trophoblast is differentiated, again this trophoblast will again be differentiated into something called as the cytotrophoblast and syncytiotrophoblast. So what is cytotrophoblast? It is a mononucleated single layer of cells, and it's surrounded by something called as a syncytiotrophoblast. So what do we mean by a syncytium? It is the continuous proliferation of the cells, and this cytotrophoblast in turn gives rise to syncytiotrophoblast, and these cells uniform. This is the cytotrophoblast, and the syncytiotrophoblast will be the outer layer, and there will not be any of the margins between the cells. So it's like a true syncytium without any inner cell borders. So the core origin of syncytiotrophoblast is nothing but the cytotrophoblast. So what one thing we have to remember is the syncytiotrophoblast cannot form by itself. It is just future of the cytotrophoblast. So all the outer cells of the cytotrophoblast is a single layer. So they get extruded into the syncytiotrophoblast. So all the nuclei will be extruded from the cytotrophoblast to the syncytiotrophoblast. So the syncytiotrophoblast does not have the ability to divide. Only the proliferation of cytotrophoblast will occur, and this will lead to the formation of syncytiotrophoblast, and that is why we cannot tell there is syncytiotrophoblasts, or it is not a plural form. It is a single layer around the cytotrophoblast. So all this occur in the pre-lacunar stage. So what happens in the pre-lacunar phase, especially between the day 5 to day 8 of the embryo? The formation of the trophoblast into the cytotrophoblast and outer layer of syncytiotrophoblast occurs. And almost at day 7, that is day 7 after fertilization, the implantation will occur. So what do we mean by implantation? The entire blastocyst is drawn into the uterine endometrium and it gets implanted, and once the implantation occurs, there is differentiation of this trophoblast into something called as cytotrophoblast, which is a mononucleated cell layer, and outer layer is called as a syncytiotrophoblast. So this is called as a pre-lacunar phase. And next comes something called as a lacunar phase. So once there is formation of the syncytial trophoblast, this is already the blastocyst is grown into the endometrium of the uterus. So if this is the uterine cavity, if this is the endometrium, it is already implanted here. So once what has happened, the fetus or the entire embryo is surrounded by the placenta. And by day 8, what happens? Day 8 after conception or after fertilization, there is vacuoles which are formed in the syncytial trophoblast. So that is why it is called as a lacunar phase. So what is the meaning of lacunar phase? These vacuoles are developing in the syncytial trophoblast and these vacuoles will enlarge and hence it is called as a lacunar uh, phase. And once these vacuoles are developing, they are again becoming larger and larger. And once they become very larger, what will happen? They will get separated from each other. So this is a layer of cytotrophoblast. This is a layer of syncytial trophoblast, and there is something called as a lacunae, which is formed between the two syncytial trophoblasts and cytotrophoblast. So that is why these are forming a type of villi. So what is this villi which is forming? There is lacunae in between the syncytial trophoblast, and that is why there is two different peaks. So multiple villi are being formed, and these gradually grow into the placental villi, and this lacunae which is formed is the future forming the intervillous space. So the future of lacunae is nothing but the intervillous space. And this intervillous space is very much essential for the maternal blood and for the diffusion of the gases and other nutrients to happen between the mother and the fetus. So hope you understood the pre-lacunar phase and the lacunar phase. So pre-lacunar phase, it is just nothing but the entire embryo is surrounded by cytotrophoblast and syncytiotropoblast, and once the entire embryo is surrounded by cytotrophoblast and syncytiotropoblast, there is formation of something called as a lacunae in between the syncytiotropoblast, and this lacunae is leading to something called as a villi like this. So this villi are formed and the outer layer is the syncytiotropoblast, whereas the inner layer is a mononucleated cytotrophoblast, and the lacunae which is formed is nothing but the intervillous space. So this is the lacunar phase. So this lacunar phase occurs between the day 8 and day 13 of fertilization. So next we have to move to the next phase. So after the prelacunar and lacunar phase, so till day 14 or day 13, comes something called as the early villus phase. So what do we mean by the early villus phase? So it is between the day 14 to day 28. So this is a very important period of after fertilization. So this is very important for the formation of the villus structure. So from the embryoblast, there is something called as the mesenchymal cells, which grow towards the placenta. So this mesenchymal cells, they grow out of the embryoblast and they form a layer beneath the syncytiotropoblast and cytotrophoblast. So this layer is called as a mesenchymal cells, and this will form something called as the secondary villi. So we have to understand what is primary villi, secondary villi and tertiary villi. So what do we know about the primary villi? It is nothing but the cytotrophoblast, which is surrounded by the syncytiotrophoblast. But whenever the mesenchymal cells are growing into this structure, so this is called as the secondary villi. And once the mesenchymal cells have grown into the layer of cytotrophoblast and syncytiotrophoblast, there is something called the tertiary villi, which is the syncytiotrophoblast, cytotrophoblast, the mesoderm, and the placenta starts developing the vasculature. So this is nothing but the vessels which are appearing, and this is something called as a tertiary villi. So the formation of primary villi, secondary villi, and tertiary villi happens in the early villus phase. So it's between day 14 and day 18, day 28, and once day 28, these tertiary villi go on proliferating, and this forms the entire villus structure, and this will occur, and these will go on differentiating, and they are staying up to the end of pregnancy, which is helping for the nourishment and the gaseous exchange for the fetus. So this is a very important concept to be understood. So as we discussed, so this is a picture which clearly makes us understand the difference between the primary, secondary, and tertiary villi. So the primary villus, there is a layer of cytotrophoblasts, and these cytotrophoblasts are mononucleate and polygonal in shape, and which is surrounded by something called as a syncytiotropoblast, with there is no internal cell boundaries, and there are multiple nuclei. And once there is intervening villus mesenchyme, which is derived from the mesenchymal tissues of the embryoblast, we call something called as a secondary villus. And then by day 18, 20, what happens? Vasculogenesis starts. So this vasculogenesis is what the hematoblastic fetal cells or the hematoblastic stem cells within mesenchyme, what it happens is hematoblastic stem cells within the mesenchyme start developing the blood cells and the endothelial cells. So the placental vasculature starts within the villus mesenchyme, and exclusively till first trimester, by the end of first trimester, like at least 11 to 12 weeks, the hematopoiesis occurs in the placenta. But after the first trimester, by second trimester till the late third trimester, the entire hematopoiesis occurs in the liver. So this connection of the vascular systems between the placenta and the embryo via the umbilical cord is happening in the lacunar phase. So this completely happens by day 35 of the fertilization, that is during the 7th week of pregnancy. There is an umbilical cord which is connecting the placenta and the fetus, and all the tertiary villi will have been completely formed. And these tertiary villi, which is a combination of cytotrophoblast, which is the layer, and outwards there is a syncytiotropoblast, and inside there is the villus mesenchyme with the placental vasculature, and these vessels are forming, and these are all dipped in the intervillus space, and in intervillus space there is the maternal blood which starts pouring, and then there is a gaseous exchange. So what happens in this early villus phase? There is formation of primary, secondary, and tertiary, and the final phase of placental development is the villus phase. So what happens in the villus phase? These tertiary villus go on branching. So they go on branching. So from one main villus tree, there grows many of the villus, and this will be going multiplying and proliferating till the end of pregnancy. And with each villus, the syncytiotrophoblastic layer will also have some microvilli also. So this helps in the exchange of nutrients or the gases between the mother and the fetus. So this is very important function of the placenta. So if you ask me, the cytotrophoblast, it's a feeder to the syncytiotrophoblast, but the functions of the placenta is many functions of the placenta are carried out by the outermost layer, that is the syncytiotrophoblast. So this syncytiotrophoblast, it is a very important layer, and it is the site of highest metabolic and endocrinal activity of the placenta. So what does it do? Does an active transport of amino acids, glucose, electrolytes such as sodium, calcium between the mother and the fetal interface. It also transports the facilitated transport of glucose. So firstly, it has active transport of electrolytes like calcium and sodium, and also amino acids, then facilitated diffusion of glucose and it also helps in diffusion of water and gases, and it forms a very important hormone. So, which is the pregnancy hormone, that is the beta HCG, which is also secreted by syncytial trophoblast, which helps in the maintenance of the pregnancy, and also the catabolism role also it has, and during the early part of the pregnancy and in the late part of the pregnancy there is also resynthesis of proteins and lipids. So in the early part of the development it is mostly catabolic, but in the late part it helps resynthesis of the proteins and, so if you ask me which is more important, cytotrophoblast or syncytiotrophoblast, I will still tell that cytotrophoblast is more important. Though the syncytiotrophoblast performs all the activities, so it is mainly for by the support of the cytotrophoblast. So what does the cytotrophoblast do? So the cytotrophoblast feeds in all the organelles, including the nuclei, organelles, nuclei, and mRNA transcripts and um, new enzyme systems, every will be transferred from the cytotrophoblast to the syncytiotrophoblast, and only cytotrophoblast is doing so much activity to the syncytiotrophoblast, and then only the syncytiotrophoblast can function like a unit and it can do all these other functions of the placenta. So mainly the metabolic activity is by the syncytiotrophoblast, but how is able to function? It is mainly by the help of cytotrophoblast. So they will always tell, if you want to shine, you want to have someone behind you. So that is how the, if the cytotrophoblast is there, only the syncytiotrophoblast can perform its function. So this is very important take home point which you all have to remember. Next is the villus stroma. So the villus stroma is usually formed by the, I already told you, it is derived from the connective tissue from the embryoblast, and also some of the macrophages. They're also called as Hofbauer cells. So these are also derived from the, some are progenitor cells from the placenta and some tell that they are derived directly from the fetus. It could be the same. Also presence of some mast cells and plasma cells in the villus stroma, but usually villus stroma is the main contributor from the embryoblast, whereas the trophoblast is the main contributor of the cytotrophoblast and the syncytiotrophoblast. So after understanding the villus systems, we have to understand the placental blood vessels. So we all know in the umbilical cord, we have two arteries and one vein. So all the blood from the fetus, which is deoxygenated, reaches the placental villi. So it comes through the umbilical arteries and these go into the villi of the arteries of the villi. And these arteries again branch into smaller arterioles and they reach the capillary bed of the placenta. And once it reaches the capillary bed of the placenta, the entire blood can get oxygenated with the transfer of oxygen from the maternal blood, which is bathed into the intervillous space in the maternal blood. And then the purified blood again enters the umbilical vein, and this umbilical vein will go, all the villus veins will drain to the umbilical vein, and the umbilical vein directly enters the cord, and then it will go into the fetal circulation. So this is all discussed in my previous video, that is the fetal circulation. So this placental blood vessels play a very important role in the gaseous exchange. So now we understood about the four phases of placental development. And so this is a picture where we have understood from implantation. So by around third week of gestation, so there is formation of villi, and this villi again get differentiated into four types. So the first formation of the tertiary villi are called as the mesenchymal villi, and then there are the immature intermediate villi, and then there is the stem cell villi, and then we go into the mature intermediate villi, and lastly there is formation of the terminal villi, which are very important. So all these are tertiary villi only. So the tertiary villi are further differentiated into these different types of villi. So what is happening here in the placenta or in the villus structure is the question. So what is happening in the mesenchymal villi? What happens? So in my next picture I will show you. But in just remember that the mesenchymal villi are usually prominent by fifth week till the 23rd week. Next comes the immature intermediate villi, and next comes the villus stabilization occurs once there is formation of villus growth, and then finally there is formation of mature intermediate villi and terminal villi after 24 to 27 weeks of gestation. So there are three phases again. It is the villus growth which occurs, then the villus stabilization which occurs, and finally the mature intermediate villi and the terminal villi help in fetal nutrition. So this slide is just to understand the pictorial representation of whatever I told earlier. So what is A? It is nothing but the mesenchymal villi. So we are seeing a layer of cytotrophoblast, and this is a layer of syncytiotrophoblast, and something we are seeing here. This is called something called as a syncytial knot. So sometimes the toxins or whatever the waste material is collected as a form of a knot, and then it is extruded into the maternal blood and it is cleared away from the maternal blood. So this is something called as a syncytial knot, which helps in the throwing away. It's like a cover to throw away the dust from the dustbin. So it is like that. So this is the syncytial knot, and this is the mesenchymal tissue, and we have the placental blood vessels. So this is a very primitive type of villi. And next we are seeing something called as the immature intermediate villi. So we are seeing here the cytotrophoblast is expanding, the mesenchymal tissue is expanding. There is also presence of macrophages and Hofbauer cells, and there is also the villi blood vessels which are present. So this is also the expansion of the mesenchymal villi. Next, coming to the third type, which is called as a large stem villus. So large stem villus is nothing but the entire villus mesenchyme has been expanded, and it has two vessels. One is the artery and the other one is the vein, and we are seeing that the cytotrophoblasts are being expanded and they are a little distance. So here the distance between the cytotrophoblast is very near. But here what is happening? There is expansion of the villus for the increase in the surface area for gaseous exchange. So that is why due to the expansion, the cytotrophoblast are seen very far away from each other. Usually the nuclei, the ratio between the cytotrophoblast and syncytiotropoblast will be around 1:9 even till term pregnancy. But we have seen that there is expansion of the villi and there is the huge stem villi which helps in the further proliferation. And next comes the D, which is called as the mature intermediate villi, and this we can see that the gaseous exchange is happening and also the cytotrophoblasts are decreasing, and because the expansion is happening there is proliferation and there is division of the villi. So if this is the villi, again it will divide and it will form the pinopodal structures. So it is proliferating and dividing, and finally we can see something called as a terminal villus. So what is a terminal villus? The syncytiotrophoblasts becomes very thin. The cytotrophoblasts are very far away and they help in the, and it becomes very thin. Why it becomes very thin? Because the fetus as it grows it requires more and more oxygen. So for more and more oxygen to happen, the should be less for the diffusion to occur very readily. So that is why the outer shell becomes more and more thin. And we have also seen the placental sinusoids which have been increased, and the villus mesenchyme is also decreased. So this helps in the close contact. So there is a close contact. So here is the intervillus space. So there is close contact between the maternal blood and the fetal blood. So this helps in the exchange of the nutrients as well as gaseous exchange. So that is why this terminal villi are very much important, and by term 40% of the villi are terminal villi. This helps in ready diffusion of the nutrients and the gases. So this is very important, and these are the different types of changes which occur in the villus architecture as the gestational age progresses. So if there is any problem in the formation of these villi, then the fetus might suffer from conditions like fetal growth restriction. So this is a picture which shows the placental bed, and here we have the umbilical cord, and which is carrying the two arteries and one vein, and this is again branching into arteries and arterioles, and this is forming the villi, and these villi have different lobes. So usually the placenta will have at least 15 to 20 lobes, and again they're divided into lobules. So this is one lobe and these are the lobules, and this contain the mesenchyme and the fetal vessels, and this is something called as the syncytiotrophoblasts, the cytotrophoblast, and the villus mesenchyme along with the vessels. And once there is the intervillus space, there is the spiral arteries which are originating, and here the diffusion occurs. So this is the entire micro picture which happens at the level of the placenta. So lastly, we have to talk something about the extravillus trophoblast. So this trophoblast is not only participating in the formation of villi, but there are also some trophoblasts which are functioning outside the villus formation. So these characteristic feature is the formation of extravillus trophoblast. So these are very much important for the development of maternal and fetal circulation. So this extravillus trophoblast is again divided into interstitial trophoblast, which is present in the connective tissue of the placenta, especially at the level of decidua bassalis, and that helps as it is like a bed. It's like a placental bed between the placenta and the myometrium. There is something called as a placental bed which is formed by the interstitial trophoblast, and some of the trophoblast go into the uterine glands, and this have something called as a histiotropic diffusion. So there is some amount of diffusion of nutrients between the trophoblast and the uterine glands. So this is called as the endoglandular trophoblast, and there are something called as endovenous trophoblast and endoarterial trophoblast, and these together are called as endovascular trophoblast. So once the so the uterine artery, it goes and it starts dividing, and it divides into radial arteries, arcuate arteries, and finally it forms something as a spiral artery, and this spiral arteries go and penetrate, and they are seen at the villi. So these are present at the intervillus space. So these spiral arteries are lined. So this is a spiral artery. This is lined by something called as a endovascular trophoblast. So this endovascular trophoblast will convert the maternal vessels from high resistance to low volume, high resistance and low volume circulation into high volume and low resistance circulation. So this is called as trophoblastic invasion. So this trophoblastic invasion up to the level of the endometrium occurs from 12 to 14 weeks, and till the inner third of the myometrium, outer third of the myometrium, it occurs till 16 weeks. So this trophoblastic invasion occurs in the by 16 weeks. It is complete, and the spiral arterioles have been converted into a low resistance high volume circulation. So this will be these vessels, the myometrial, these spiral arterioles, the tunica media will be replaced by the endovascular trophoblast, and that is why they will not be responsive to the systemic vasoconstrictors like angiotensin. So that is why they do not constrict readily. Instead, they will be like a large pipes which are supplying the blood from the mother to the fetus. So there it will be in the intervillus space, and when these villi are bathed in the intervillus space, the selective diffusion of all the nutrients and gases occurs. So if there is any defect in the trophoblastic invasion, this might lead to something called as preeclampsia syndrome. So that again is divided into stage 1 and stage 2 disease of preeclampsia syndrome, which is a topic for other It's a topic for another day for discussion. But today you are just understand that these spiral arterioles have to be remodeled, and if there is any defect or deficiency in the trophoblastic invasion, this might lead to preeclampsia, and further it might also lead to fetal growth restriction consecutively. So this endovascular trophoblasts are very much important in the formation of the low resistance and high volume circulation. And there are some trophoblasts which also go into the endolymphatic, uterine lymphatic vessels, but the role has not been established. So these are the functions of the extravillous trophoblasts. So now we have understood that the trophoblast has been differentiated into both syncytial and cytotrophoblast and also the extravillous trophoblast, and we have understood the function of the same. And finally, when we get the placenta in our hand, so now we can understand which side is the fetal side and which side is the maternal side. So the fetal side is completely covered by the fetal membranes. So this amniotic membranes are also from the embryoblast, and also the umbilical cord also is arising from the embryoblast. So this is side from the fetus, because from the fetus there is the embryoblast is giving the umbilical cord, the amnion, the chorion, and the villus stroma is formed by the fetal side. But on the maternal side, there is the complete lobes of the placenta. So these lobes of placenta, around 15 to 20 lobes will be there, and this are also called as the maternal cotyledons. So this is the villi which have been branched, and this is the formation by the trophoblastic tissue which is entirely forming the placenta and the villus structure, and this will be in direct contact with the uterus. So when it is in direct contact of the uterus, the interstitial trophoblast will act like the placental bed. And from there, so this is in contact with the uterus. So if this is the uterus, if this is the placenta, so this maternal side is in contact with the uterus. But this is the fetal side. So this is the fetal side which is contact in the fetus. And here there is spiral arterioles which are coming, and there is maternal fetal interface. So in Toto, this class explains the development of placenta. I hope you have understood the phases of placenta, the types of villi which are forming in each phase of the development of placenta, and also understand what is the role of extravillous trophoblast. With this, I will conclude my today's session, and in this placental series, we will be covering more and more interesting concepts about placenta. Thank you. Happy learning. And you can watch this video and understand the development of placenta. Thank you. Have a nice day.